There is an important organelle in the cell that is almost always undermined in high school biology classes. This is the cytoskeleton. The cytoskeleton is an interconnected complex network of filaments that exists in all cells, prokaryotic and eukaryotic. It serves to create cell structure and structural integrity, allows for cell migration, plays a role in cell signaling, intercellular transport, endocytosis, chromosome segregation, and cytokinesis, the last two being part of cell division, supports an entire tissue's integrity, forms other additional structures like flagella and cilia, and so much more depending on the type and function of the cell. This will be a four-part series, the first three of which will explore one of the three main components in the eukaryotic cytoskeleton organelle: microfilaments, intermediate filaments, and microtubules. The last will explore the other parts of the cytoskeleton and summarize all of the videos. This video will talk about microfilament and actin creation. Microfilaments, also known as actin filaments, are the smallest structure out of the three in terms of diameter, approximately 7 nanometers. They are constituted by a variety of actins, a type of protein that is very abundant in cells. There are mainly three types of actins constituting microfilaments: the alpha actins, which are found in contractile microfilaments; beta actins, which are used by microfilaments that play a role in cell mobility; and gamma actins, which are found in microfilaments withstanding stress. All of these actin monomers are called globular actins because of their roughly spherical shape, and they polymerize to form filamentous actin, which is the basis of microfilaments. These G-actins, made up of 375 amino acids, are rotated 166 degrees in the filament, giving the microfilament the appearance of a double-stranded helix. There are three main steps in actin polymerization: the nucleation phase, the elongation phase, and the steady-state phase. In vivo research finds that the formation of F-actins starts off with some kind of nucleating factor, such as the ARP2-3 complex. This protein complex can mimic a G-actin dimer to cause the nucleation of a G-actin trimer. Trimers and chains with a higher number of monomers are relatively stable, and from here, the elongation phase begins. In vitro research finds that because of the high ion concentration environment present in cells, more monomers can spontaneously attach to either side of the trimer. Note that the G-actin monomers attach 5 to 10 times more frequently to the plus end, also called the barred end, of the G-actin trimer, as shown here. Additionally, ATP can bind to the activation sites in the G-actin monomers to expedite the process. Conversely, if the ATP is hydrolyzed into ADP, the process will slow down. This is the most common way that microfilament growth is regulated. Thymosin beta-4, a peptide, and profilin, a protein, are the two main regulators of F-actin creation. Thymosin beta-4 can bind to the G-actins and act as a buffer, restricting the binding of the affected G-actin monomer to the chain. This happens through the equilibrium equation here, where a higher concentration thymosin beta-4 would cause more G-actins to bind with the thymosin beta-4, correspondingly decreasing the concentration of F-actins. Profilin is one postulated to bind to the plus end of a G-actin monomer to allow for the minus end, also called the pointed end, to bind to the more binding active plus side of a growing actin chain. 2. Involved with cell signaling near the cell membrane, suggesting that it is especially crucial to actin polymerization. And 3. Is the only protein that binds to actin that allows the exchange of nucleotides between the actin, for example, ADP to ATP. This exchange of nucleotides allows for a more ready bonding and a stronger bond compared to the G-actin with ADP. One thing to mention here is that while the F-actins can resist a lot of stress and pressure, they have relatively weak non-covalent lateral and longitudinal bonds, with free energies of -1.9 to negative 3.4 kilocalories per mole, and negative 4.0 to negative 5.6 kilocalories per mole, compared to the relatively strong covalent phosphodiester bonds supporting the monomers with a free energy excluding the hydrolyzation energy of ATP to AMP and 2-phosphates calories per mole, allowing the microfilaments to easily conform and change shape in response to environmental stresses. Sources are here. Since profilin also causes these G-actins with ATP to mostly bond to the plus end of the G-actin chain, the ADP G-actins on the minus side could lose its bond with the chain, receive an ATP through the profilin, then proceed on to the positive side. This process is known as treadmilling since it looks like the G-actins on the negative side are disappearing and reappearing on the positive side. Other proteins like cofilin and gelsilin can sever the chains by binding to them, causing conformations, which then lead to destabilization of the bonds between the monomers, and others can act as caps by basically binding to one end of the chain. All of this happens so that the G-actins can form F-actins. F-actins can form either actin bundles and actin networks as shown here. These two types form microtubules and act to disperse stress. The next video will talk about how the microfilament moves and what functions it has. This is post-recording, and there were some things I want to clarify. There were some errors with the bonds, and basically the main point was that while the monomers, the G-actin monomers themselves, do not have strong bonds that hold them together, nor do they have strong lateral bonds that holds the neighbors together. Collectively, they can spread out that force and smooth it out, unlike things like DNA or RNA, which already have strong bonds. For example, the phosphodiester bonds between the ribose and the phosphate group inside RNA is an example of a strong bond that would last. So, thanks for watching.